water because they are higher concentration of water inside than outside. So they will shrivel, they will crenate, and cells will collapse. So that condition is called crenation when we talk about animal cells, and the same condition in case of plant cell is called plasmolysis. It is plasmolysis, so it's read as plasmolysis. It means the loss of plasma. Plasma is cytoplasm in case of cells. So the cells in case of plants also shrivel. So have you ever eaten any plant that is shriveled? Have you ever eaten a salad that is not really crunchy? So that is because of plasmolysis, because cells have lost their water, not because of actually this process, but they have lost water because Walmart or wherever you buy your salad from, they did not really put that mist on them. So they lost water and nobody really buys that salad. But that is because of the shriveling or loss of water in them. This also shows the three conditions of isotonic, hypotonic, and hypertonic. This is hypotonic in the middle, and this is hypertonic. When the solution is isotonic, there's no gain or loss of water. When it is hypotonic solution, then the cells will gain water from outside, and then they will go through that process of bursting. In case of plant cells, actually they don't burst because they have cell wall. So cell wall does not allow cells to burst. They will expand, but they will be restricted with the help of this cell wall that will contain them. In this case here, in hypertonic, then cells will lose their moisture, so they will shrivel. So in this case, this is a shriveled cell, and in case of animal cells, since they don't have cell wall around them, so they will just kind of collapse and become very small. Yes? You have a question?
The percentage you don't put in
So if somebody does not mention, for example, if you found out that somebody says 10% solution, then that means this is just salt. This is the concentration of the salt. Because by default, they don't say that this is 90% water. It just says that 10% solution means 10% of the salt in it. No, so if somebody says it is 70% water, then that's the concentration of the water. So if somebody says that I have a solution that is 70% water, so what does that mean to you? It means it has 30% of the salt, right? So if I say that I have a 30% solution of sodium chloride, so automatically you will calculate that it has 70% of water in it. So whenever there is no mention of solvent, that means by default it is the concentration of the solute. That the concentration is the salt, not the solvent. So for example here I think there is a mention of 0.9% somewhere, probably not. Okay, so 0.9% is the solution which is isotonic. So that's what is used in those drips. If you're dehydrated or if you have certain conditions, hydration is done with the help of those type of solutions. Any other questions?
That's why you see on the bag. So you get an IV, and it says 0.9 sodium. That's just saying it's 10%.
That's right.
Of the solute.
Yes, it's only 1%. 1%. 0.9% means it's almost 1%. So 0.9% saline is that much sodium chloride, but it also has something else in it too. They contain, I think, some sort of carbohydrates in there too.
Yeah, sure.
Yeah, dextrose or something like that. Depends on what it is. Okay, so the other type of transport is facilitated transport. Facilitated transport means it is done with the help of proteins, which are embedded in the membrane. So things move from higher concentration towards lower concentration through specific proteins. Proteins enable them to be carried from higher concentration towards lower concentration. In this case also, there is no involvement, there is no expense of energy also. So it is going from higher concentration towards lower concentration through those type of channels which are specific for a type of a substance. For example, glucose channels. glucose is needed inside the cell, so glucose crosses the membrane through its special gate, which is these type of proteins. So it has a special status. It's a VIP status. Water has a VIP status. Glucose has a VIP status also. They have their own gates. Nobody else actually goes through those gates. So they bind to the protein, and then proteins actually move them to the other side. Active transport is at the expense of energy. It is called active because energy is required. ATP is required to carry things from one side to the other. So this is a special type of transport in which energy is required. This type of transport is done in the cases where things are moved against their concentration gradient. So far what we have studied through diffusion, osmosis, or facilitated transport, those are all in favor of their concentration gradients. Things are moving from higher towards lower concentration. That's why energy is not required. For example, when you're going downhill, you don't really require energy for that purpose. When you're going uphill against the concentration, against the gravity, then at that time you need energy. For a person like myself, I will start hopping, because I'm not used to exercise. So when I'm going uphill, I will start to struggle because I don't have that energy in my muscles that are needed. So the same thing is happening in this case also. When things are going from lower concentration towards higher concentration, then you have to input energy at that time. You have to provide more energy to enable this type of a transport. This is done in cases where things which are really important inside the cells are moved inside the cell. Like, for example, this pump, which is a protein that pushes some of the ions like sodium outside the cell and brings potassium inside the cell. Sodium is not good inside the cell, potassium is good. So, and even though the concentration of potassium may be higher inside the cell, but it is still brought inside the cell because it is a good thing. The cell wants to keep potassium inside. So this is that example of sodium-potassium pump. Sodium outside and potassium inside is the theme in this case of this pump. So energy is required. For example, at this time, this channel is open, which is a channel for both for sodium as well as for potassium. So when this channel is open at time 0, these sodium ions will bind to its own specific site inside this protein. So there are 3 sites for this sodium ions to be attached here. So they will attach here. Now it is closed from the other side. It is not open. So those sodium ions cannot really go through this channel. They will bind here and then they'll sit here, wait for the energy when energy is available. When energy is available in the form of ATP molecule, it will bind here, and the binding of this ATP molecule will change the structure of this protein. It will close it from this side and open it from this side, which is shown here. So when it opens here, because of the energy, the molecule of energy which is hydrolyzed, so that energy is made available. So that energy will open the channel to the other side of this membrane. So when it opens on the other side of the membrane, guess what happens at that time? Now it is open to the outside. These ions that are present on the outside, like potassium, they will bind to their own side on this pump, on this protein. Pump means it's a protein, it's pumping things. So these two bind here, and their binding help them to actually be moved to the other side. Then in the next event, this phosphate group, which is added here, is removed from it. So its removal will bring it back to the same position as that position over there. So the removal of this potassium will change the structure of the protein again. So it will open on this side again. When it opens on this side, those ions that got attached here, they will be released on the inside of the cell. So it has actually carried out 2 different things at the same time. Got rid of all the things that it does not need and brought in the things that are needed inside. And both of those are going against the concentration gradient. So it kind of killed 2 birds with 1 stone here. With only 1 ATP molecule, it got rid of sodium ions and brought in the potassium ions inside the cell. The same is the condition in case of exocytosis and endocytosis. Exocytosis is to bring things inside. There are certain categories or certain types of endocytosis when things are brought in. One is what is followed by our immune cells. Our immune cells can engulf other cells. They can eat other cells. That process is called phagocytosis. Phagocytosis means cells eating other cells. So they engulf other cells. For example, some cells which are bacterial cells, or cells which are dying, or cells which are have mutated in somehow, are removed from the body with the help of this process, which is called phagocytosis. Pinocytosis is a small mechanism in which the cells eat things from outside of the cells. If they're present on the outside, they enclose them, they bring them inside. Okay. Just very quickly on ECM, extracellular matrix. Extracellular matrix is the cement which is present on the outside of the cell. Cells are attached to that matrix. It is called extracellular, means it's on the outside of the cell. It is extracellular matrix on the outside of the cell. It is made of some proteins and some polysaccharides, carbohydrates. And it is a glue, it is an attachment point, it is cement that is present on the outside of the cell. And these are the proteins that are present in that kind of attachment point on the outside. So these help things to be attached inside to the outside, and things which are present on the from the outside of the cell as well. So things which are present in the inside in the form of, so this is the outside of the cell, this is the inside. These cytoskeleton proteins, like if you remember this, actin filaments, they are attached to these proteins to the outside and are anchored on the outside of the cell. These are those junctions. These are the three types of structures the cells make to be held together with each other. Adhesion junctions are the ones which are present between cytoskeletons of the two cells. These are the two cells which are attached through these junctions with each other. So they are anchored like that. They are kind of attached on those points. And then the tight junctions are the ones which are actually present on the top of the cells. Like this is a cell, this is another cell. So these are present here. They basically choke the top part of the cell in such a way that things don't fall through this area. So especially, for example, there is food flowing in our digestive system. The food which is present on this side will not actually be able to go through these tight junctions here. Yes, if it goes through here, there is no way of recovering that food. So in order to protect those cells, to have food in between them, these tight junctions are in the form of proteins. Then the gap junctions are connections between cells. Gap junctions are holes that are present between the two cells, which are adjacent cells with each other, so that helps cells to send their signal to each other. If they have to borrow a cup of sugar or something, they can actually do that from these channels, through these windows also. So they can actually pass their signal from here to here, or from here to here. So they actually are direct connections between two cells. The same type of system in case of plants is called plasmodesmata. Plasmodesmata are these type of channels, which we saw in the previous slide also. Instead of calling them gap junctions, they are called plasmodesmata in case of plant cells. All right, so on Monday, so we have a test.